Senior High School on Air.
Senior High School on Air. The frontline of quality and excellent learning.
Excellent learning. You have all the best of the best.
Best of the best of the best.
Magandang umaga, handa na ba kayong matuto?
Handa na po.
Reading and writing.
Pagbasa at pagsusuri ng teksto tungo sa pananaliksik.
Practical research 1.
English for academic and professional purposes.
Variability and statistics.
General Biology 2.
Disciplines and ideas in applied social sciences.
Cookery and more.
Senior High School, best of the best.
Best of the best of the best.
Sam Kian, a science writer, once said, "The genes are like story, and DNA is the language that the story written in." Good day, senior high school students! Another quarter has ended, yet still, your eagerness to learn keeps on levitating. You are currently listening to Radyo Eskwela sa Isabela for General Biology 2, sharing with you invigorating lessons that will surely spice up your radio-based learning experience. Let us embrace the new normal and spread a positive outlook in studying. I am your science buddy, Teacher Erwin. Another quarter, another learning journey. I know you are enthused to open another chapter of this school year. Are you excited? I'm sure you do. So, I hope that everyone is ready. I expect your active participation and engagement for today's episode. Let's go! Before we begin with our discussion, let us check what you know about our new topic for today. Answer the questions that I will ask. Relax. Just answer what you know. Let's begin. In genetic engineering, what is transferred from one organism to another to produce a genetically modified organism? Absolutely. The answer is deoxyribonucleic acid, or DNA. What is the name of the structure found in bacterial cells into which genes may be inserted? You got it right. It's plasmid. In genetic engineering, what is used to cut the required gene out of the chromosome? Very good. Genes. The process of making changes in the DNA code of a living organism is called? Correct. The answer is genetic engineering. Job well done, my dear students. I believe you are now ready for a new topic. Let's continue after the short break.
Good morning, Miss Santos. Oh, good morning too, Mrs. Cruz. What can I do for you? I just want to ask, ma'am, are we the ones to teach our children at home during this pandemic? As we have already given the modules, we encourage you to guide your child for them to complete all the given activities. Do not worry because instructions are clearly provided in their self-learning materials. Just provide your child the necessary adult supervision while doing the tasks. If you have queries, you can always reach me. We cannot do anything but to follow the new normal way of learning. Worry not, if everyone stays at home, COVID-19 will go away. Thank you, Mrs. Santos. I promise to guide my child the best way I can. Have a good day, Mom. Thank you for your cooperation, Mrs. Cruz. Likewise, keep safe. In DepEd, modular learning under the new normal is one of the modalities. Sulong Edukalidad. We recover as one. A friendly reminder from the Department of Education and this station.
What is the significance of mitosis? You're right. We studied that mitosis is an equational cell division that produces daughter cells which are identical or clones of the original mother cell. This ensures that every cell of the body has the same genetic content, that is, chromosome number. To make this possible, cells have to duplicate their genetic material, which is primarily DNA. Imagine how many cells a typical mature human contains. Do you know that they all came from just one fertilized egg cell? A zygote goes through millions of generations of cell division to become just the one person that a learner is. Even until now, cells in an individual are still dividing. What examples of tissues in the body are undergoing cell division? Yes, skin and blood cells. I am sure you already have an idea about our topic for today. That's right, it's all about recombinant DNA. At the end of this episode, you are expected to outline the processes involved in genetic engineering and discuss the applications of recombinant DNA. Deoxyribonucleic acid may be a large biomolecule that contains the entire genetic information for an organism. Every cell of living organism and lots of viruses contains DNA. The fundamental building block of a DNA molecule is termed a nucleotide, and one strand of DNA may contain billions of nucleotides. Wow! Amazing, right? Because DNA contains instruction for an organism to form several different proteins. It is useful to define another subunit of DNA called genes. Each gene may be a small segment of DNA that contains a group of instructions for an organism to form one protein. One organism may have thousands of virus genes. Together, the complete set of genes for an organism is named its genome. The technique, genetic engineering, allows scientists to provide more complex solutions to problems within the society. The term biotechnology came from the words biology and technology. It had been coined within the 1970s when the primary genetically engineered bacteria were reported. Since then, biotechnology has often been related to gene engineering, specifically within the development of genetically engineered microorganisms. But what is genetic engineering? By definition, genetic engineering is the direct altering of an organism's genome. This can be achieved through manipulation of the DNA. Doing this can be possible because DNA is sort of a universal language. All DNA for all organisms is formed of the identical nucleotide building blocks. Let us discuss the process for genetic engineering. The process for genetic engineering begins the identical for any organism being modified. First, choose an organism that contains a desirable gene. Second, extract the whole DNA from the organism. Third, remove this gene from the remainder of the DNA. A way to try this can be by employing a restriction enzyme. These enzymes seek for specific nucleotide sequences where they will cut the DNA by breaking the bonds at this location. Fourth, insert the new gene to an organism's DNA. What do you think will be the last step? Curious? Let's continue. When modifying bacteria, the foremost common method for this final step is to feature the isolated gene to a plasmid, a circular piece of DNA employed by bacteria. This is often done by cutting the plasmid with the identical restricted enzyme that was needed to remove the gene from the initial DNA. The new gene can now be inserted into this opening within the plasmid, and also the DNA are often bonded back together using another enzyme called ligase. During this case, the recombinant plasmid is additionally named as bacterial artificial chromosome. Do you know that once the recombinant DNA has been built, it is passed to the organism to be modified. Yes, you heard it right. If modifying bacteria, this process is kind of simple. The plasmid may be easily inserted into the bacteria where the bacteria treat it as their own DNA. For plant modification, certain bacteria like bacteria species is also used because these bacteria permit their plasmids to be passed to the plant's DNA. In application and economic, the quantity of utilizations for hereditary building is expanding as increasingly more is found about the genomes of various living beings. Some of fascinating or outstanding application zones are depicted beneath. Various different qualities can be joined with yields to create alluring properties. For example, herbicide, dry season, freeze or disease resistance, higher yield, faster development, improved sustenance, and longer span of usability. Are you still with me? That's good to know, because we still have a lot to discuss. Genetic engineering includes the utilization of atomic strategies to alter the attributes of an objective living being. The adjustment of attributes may include presentation of new attributes into a creature, upgrade of a current attribute by expanding the statement of the wanted quality, and upgrade of a current attribute by accepting the restraint of the wanted quality's demeanor. How about in recombinant DNA? Let's dig deeper. An overall layout of recombinant DNA might be given as follows: cutting or cleavage of DNA by restriction enzymes, choice of a suitable vector or vehicle which would proliferate the recombinant DNA. Example given: runabout plasmid in microbes with an unfamiliar quality of intrigue. Ligation of the quality of intrigue from creature with the vector cut bacterial plasmid. Move of the recombinant plasmid into a host cell that would convey out replication to make enormous duplicates of the recombined plasmid. Determination cycle to screen which cells really contain the quality of intrigue, and sequencing of the quality to discover the essential structure of the protein. Did you know that there are few strategies to screen recombinant cells? Yes, they are the determination of plasmid DNA containing cells, choice of changed cells with the ideal quality, and PCR identification of plasmid DNA. This time, let us talk about genetically modified organisms or GMOs. Do you have any prior knowledge about it? Let's find out. With the capacity to embed quality arrangements comes the chance of giving new characteristics to these objective creatures. This has permitted the advancement of GMOs. A portion of these hereditary changes guarantee higher item yield for their objectives. The flavor-saver or flavor-saver tomato was the primary hereditarily altered creature that was authorized for human utilization. The characteristic change in this tomato is its aging cycle, a quality for a compound that causes the debasement of gelatin in the cell dividers. For example, polygalacturonase, typically mellowed organic product as it matures. BT corn was created to consolidate the creation of a poison. For example, BT endotoxin from Bacillus thuringiensis in corn plants. These poisons bring about the passing of nuances that feed on these plants like the corn drill hatchlings. A similar innovation has been applied in the Philippines for the advancement of BT eggplant. To further understand our topic, let's have a short activity. Say yes if the statement is true and no if false. Understood? Okay, let's begin. Choosing an organism that contains a desirable gene is the first step in genetic engineering. Yes or no? Correct. It's a yes. Number 2. Genetic engineering is not the direct altering of an organism's DNA. Is it yes or no? Absolutely. The answer is no. Let's have number 3. Deoxyribonucleic acid may be a large biomolecule that contains the entire genetic information for an organism. You got it right. It is yes. Number 4. When modifying bacteria, the foremost common method for this final step is to feature the isolated gene to a plasmid. Great! It's a yes. Good job, my dear students. Always remember these points. Deoxyribonucleic acid may be a large biomolecule that contains the entire genetic information for an organism. Every cell of living organism and lots of viruses contains DNA. The fundamental building block of a DNA molecule is termed a nucleotide, and one strand of DNA may contain billions of nucleotides. The determination of plasmid DNA containing cells, choice of changed cells with the ideal quality, and PCR identification of plasmid DNA.
They say knowledge is power in this world full of devour. Be prepared in mind and never get reckless. We'll conquer the world. Let's be educated. Education is indeed a key to success. Education must continue despite of the pandemic. A simple reminder from the Department of Education and this station.
It is now time to assess what you've learned through a short quiz. Kindly prepare your quiz notebook and write the date today. Your quiz has 2 parts. For the first part, it is a multiple choice type of test. It consists of 10 items only. For the second part, it is outlining the process of genetic engineering. Are you ready? Of course you are. Let's start. For the part 1 of your quiz, let's have the multiple choice type of test. Number 1. One advantage of genetic engineering for agriculture is blank. A. Cloning plants. B. Curing genetic diseases. C. Increasing yield. Or D. Mass production of plants. Number 2. Researchers have been working on cloning animals such as sheep. Which result is more likely a negative result of cloning? A. Low rate of genetic diseases. B. Delayed aging. C. Development of extra organs. Or D. Premature death. Let's have number 3. Which of the following is not associated with genetic engineering? A. Cloning. B. Expression. C. Transformation. Or D. Translation. Number 4. Which of the following is the salient feature of genetically modified organisms? A. Are always harmful. B. Are always microorganisms. C. Arise naturally. Or D. May be beneficial. And for number 5, what is recombinant DNA? A. DNA that has been sequenced. B. DNA that causes genetic disorders. C. DNA which has been changed over generations by natural selection. Or D. Molecules of DNA from 2 different species that are inserted into a host organism to produce new genetic combinations. Let's continue. Number 6. Which of the following is not an issue associated with genetic engineering? A. Introduction of toxins into the human body. B. increased exposure to vitamins and minerals. C. The development of new allergies. Or letter D. Pests developing genetic resistance. Number 7. In agriculture, how has genetic engineering helped to manage unwanted insects? A. By developing new insecticides. B. By inserting the genes for bacteria into the crop. C. By using genetically modified chemicals. Or letter D. By deleting gene sequences in the crop. Let's proceed to number 8. Which of the following statements best describes genetic engineering? A. It is an alteration in the genetic makeup of an organism by inserting or deleting in specific pieces of DNA. B. It is an alteration in the genetic makeup of an organism by deleting or changing specific pieces of DNA. C. It is an alteration in the genetic makeup of an organism by inserting, deleting, or changing specific pieces of DNA. Or letter D, it involves the alteration in the genetic makeup of an organism through inserting, deleting, or changing specific pieces of DNA. Number 9. What is the ultimate source of genetic variability? A. Evolution. B. Genetic drift. C. Haplotype. Or D. Mutations. And last but not the least, which of the following steps is not essential in producing recombinant DNA? A. Read the sequences of bases in of DNA. B. Cut out a piece of DNA from a DNA molecule. C. Use a restriction enzyme to cut DNA and from sticky ends. Or D. Insert a piece of DNA from one organism into the DNA of another organism. Now, let's have part 2 of your quiz. On a piece of paper, outline the processes involved in genetic engineering using a graphic organizer. I will give you 2 minutes to finish your quiz. Ready? Time starts now.
Tayo ay mag-aral sa loob ng ating tahanan. Sa radyo ay mag-tune lang, tiyak ika'y may matututunan. Pagkat sa gitna ng pandemya, tayo pa rin ay magkaisa upang masigurong bawat bata ay pumapasa. Halina't matuto gamit ang ating radyo, pakinggan at unawain, aralin na ituturo. Patuloy ang edukasyon para sa ating henerasyon. Sa daan ng pagkatuto ay walang maiiwan. Kaya halina sa Radyo Eskwela. Pagkat kayo'y kinabukasan, ang pag-asa ng ating bayan. Isa pusong pagkatuto nang matupad ang pangarap mo. Pagkat sa gitna ng pandemya, tayo pa rin ay magkaisa upang masigurong bawat bata ay bumabasa. Halina't matuto gamit ang
And that's it. We are done with your quiz. It's quite easy, isn't it? To know your scores, tune in on our next episode for our checking. Okay? Don't forget to pass your quiz form on time. Reminder, let your parents or guardian submit your requirements following the safety protocols. For any questions and clarification, don't hesitate to contact your teacher. Thank you for joining me today. It has been fun knowing that you enjoyed and learned a lot. Congratulations for a job well done. Another episode has ended, but the challenge to strive and study more is still on. I'm rooting for you. Let us give credits to Miss Sharon C. Terayo for this script and the whole RBI production team. I am your science buddy, Teacher Erwin. Until next time for another fruitful learning. Always remember, change is the end result of all true learning. Have a good day!
Patuloy ang edukasyon para sa ating henerasyon.
Sa daan ng pagkatuto ay walang may iwan.
Kaya halina sa Radyo Eskwela!